Hello folks. In this video, I want to talk to you about receptors of the somatic nervous system and in terms of what type of stimuli those receptors are uh, being activated by, or what type of stimuli is turning on or turning up or down the response of those receptors, and whether those receptors are always sending information to the nervous system, or whether they're only sending it when sensory input or sensory stimulus intensity is changing. So we'll think about those two things as we look at these different receptors. So receptors vary in a couple of ways, and just a little background for this is that we're going to remember that some receptors are very specific to a particular type of stimuli, like mechanical stimuli or chemical stimuli. In other words, that mechanical deformation of the plasma membrane might result in the opening or closing of ion channels and the stimulation of that receptor. And in other cases, the binding of some ligand might result in the opening or closing of ion channels, and then that would alter uh, the membrane potential of the receptor. So some are specific, some are general. Um, all of them have a specific receptive field, and you'll remember we looked at receptive field in the last lab, where we did our 2-point discrimination test, and we determined about how large the receptive field was. Here's my sorry, there's my little uh, uh, paperclip, right? Remember that we looked at, for example, the fingertip, and we figured out how far apart we had to get before the fingertip could discern that there were actually two prongs there, and that was very different from when we did the arm and figured out how far apart we had to get. I'm still not feeling the difference before we actually felt that there were two different points of contact, right? That tells us about the receptive field of the neuron. When receptive fields are very small, then neurons tend to be densely uh, populating the area, or densely packed, and that's why we have such an ability to determine such fine differences in pressure points in our fingertip, because receptive fields are very, very small, right? And here's my receptive field here. And so there's a neuron right next to it with another receptive field that size and all the way around, versus in the forearm, right, where the receptive field, there I can actually feel two pinpricks. The receptive field is very large. The receptive field of any single neuron is very large, and therefore all the receptive fields of neurons around it are also very large, right? So this, and we saw that this just depends on, how important this particular part of our body is to sensing mechanical stimuli. So this is true of all receptors. They have receptive fields that are large, small, medium, depending on how much area they need to cover. Then we also have sensors that differ in what they respond to, what types of stimuli they respond to. The one kind of weird one are these ones we call nociceptors, because what it responds to is stimuli that's painful. Well, what is that? That's not a thing, right? Pain is just a perception that we develop in our brain. It's not actually a physical thing that you can measure, right? So nociceptors are just neurons that respond to stimuli that we ought to perceive as painful, right? So very intense heat, or very intense cold, or the shearing of, the cutting of a neuron, right? Or the and when an acid maybe is dumped on the skin and starts deteriorating the cells and disintegrating the cells, that those are examples of types of pain that we need to, or types of stimuli that we need to perceive as painful, so that we can withdraw the body from those stimuli before it gets severely damaged or results in death, right? But what I just described are not stimuli. What I just described are like things that are noxious for you, right? So nociceptors some nociceptors can respond to any of these types of stimuli, to temperature, extreme physical deformation or breaking, or chemical cues, right? And some nociceptors respond to just one or the other. When we talk about things like intense pain, burning, we've learned that when we talk about when people talk about an intense pain or a burning pain, that perception, it's because all three types of nociceptors are activated. Those that respond to temperature, those respond to physical changes to the plasma membrane, and those that respond to chemical cues, and even nociceptors that respond to all three of these. And we'll take a look at actually one of these nociceptors in the special senses. Okay, so that's a weird one, but then the rest kind of makes sense. There are thermoreceptors. They respond to changes in temperature, right? And that's just because changes in temperature change the shape of the plasma membrane or proteins in the plasma membrane that change the permeability of the cell to ions, particular ions. There's mechanoreceptors, those that respond to some kind of physical change or deformation or pressure on the membrane. And there's chemoreceptors, those that respond to some ligand binding to either to a channel, in the case of like a direct stimulation, or bind to some other receptor that kicks off some intracellular pathway that involves opening an ion channel. So in all these different cases, and then there's one weird one that's that are, and it's not a general sensor. It's not one of our sensors of our general senses, but it's a special sense, and that occurs in the eye, and that's really responding to electromagnetic radiation. But that's kind of the one weird one. But these are very typical of our general senses, these types of receptors responding to these types of stimuli. And then, of course, nociceptors are this odd one. Okay, so the way that sensors respond and their pattern of action potential firing can tell us a lot about the stimulus that we're receiving, whether it's changing or not changing, how intense it is, and how long it's lasting, right? So this is called sensory coding. And we do that we have two different types of receptors, or in terms of sensory coding. And they vary in their activity. Tonic receptors are always active. They're said to be constitutively active, right? They're always firing action potentials. And these receptors uh, are firing action potentials at some default frequency, right? Maybe every 5 seconds or something. Now, when a stimulus intensity increases, then perhaps the firing of those action potentials also increases to match that. If the stimulus intensity decreases, the frequency of action potentials would drop below the default action potential firing or rate or firing frequency. And these receptors are called slow adapting, in that they don't adapt to the stimulus. So if a stimulus is there, right, they are firing. What if a stimulus is there for a really long time? They're still firing. They're still sending action potentials to the nervous system and telling us about that stimulus, right? Because it's just one of those that's important for us to know about. So, for example, nociceptors behave in this way. They're tonically active. If there's ever some sort of stimulus that is causing us damage such that we should interpret it as pain and remove ourselves from it, we need to know about it, right? So they're always firing. The other type are phasic receptors. They're normally not firing action potentials at all. They're inactive. But whenever there's a change in the stimulus intensity, then they start firing, right? So whether that change is an increase or it's a decrease, they will fire during those periods. The frequency indicates the rate of change, right? I mean, it also, in a sense, indicates the intensity. So, for example, if we had a very steep rate of change of the intensity of the stimulus intensity, they would fire very rapidly, right? And if we had kind of a slow degradation of the stimulus intensity, they would fire less rapidly, right? So they're really firing based on the rate of change of the stimulus. These are called fast adapting receptors in that they pretty quickly adapt um, to a stimulus. They stop firing as soon as the stimulus stops changing. And even if this stimulus is changing very slowly in one direction or another, they tend to adapt and stop firing because it ceases to be a relevant stimulus to think about. So thermoreceptors, general thermoreceptors, not those that are detecting extreme heat or cold, but general thermoreceptors do this, right? When you first go from a warm room to a cold room, you really notice it, right? But after a few minutes, you basically don't notice or can't feel that difference from going from warm to cold. And this is because these are phasic receptors that have adapted very quickly. Now, this is peripheral adaptation at the at the place where the stimulus is turning on or the receptor. There's also central adaptation, right? So you remember that all sensory stimuli gets funneled to the thalamus. And so, of course, we're talking here about the somatic nervous system. So all somatic sensory information first runs through the thalamus before it gets projected to some area of the cerebrum for us to develop a perception about it. So this is another place that we can edit out this information. Let's say, for example, I mean, there's different types of pain. There's chronic pain, there's acute pain. Acute pain tends to be stuff we really need to pay attention to because it's happening and it's happening right now, and it hasn't been happening for before, and typically it's an intense pain, so we should pay attention to it. But then a lot of chronic pain is like it's just around, it's hanging around. After a while, you may not even notice chronic pain, right? Or it's such a low level that it's not critical that you're always aware of it, right? In fact, if you were, it would probably drive you mad. So in that case, there's central adaptation. The thalamus just says, okay, this is a pain that's not too bad. It's here all the time. Let's not think about it anymore because it's not going to hurt us, right? Okay. So let me just show you very quickly uh, what that looks like, what those receptors would look like. I'm going to share the sketchbook with everyone. You'll see here I've put axes we're familiar with, millivolts on this axis, so we're measuring membrane potential. Time on this axis, but note that we're not in milliseconds, we're in seconds here. That's so I can put many action potentials on here. So we're in seconds. I'll just indicate an action potential by a vertical line. And then here's intensity on this axis. This is going to be stimulus intensity. And we don't need to put values on it, but we can just say, maybe we're at 0 and it's above or below 0 is stimulus intensity. Right? So let me draw some stimulus on here. So here's our stimulus intensity right here, right? We'll just say there's a flat, right? Flat stimulus intensity. And then we'll go up. So an increase, so higher stimulus intensity. And then let's say we'll have this kind of long, slow drop in intensity. And there we are again at flat or default, or let's call this basically 0, right? This is just no stimulus in any particular direction at all, right? So what would a tonic receptor do? Tonic receptor, here we are in pink. Well, a tonic receptor is always firing action potentials, right? Whether there is any stimulus intensity or not, right? So we have 0, we have no stimulus here, right? So let's see, I'm going to draw some vertical lines here to indicate action potentials, right? So here's our action potential. There's one, and there's another, right? And they're at this fairly low rate, but now we get some stimulus here. So we start to get action potentials firing, right? And that was so steep that I wasn't really able to draw much of a difference in action potential frequency occurring along that line, that really steep increase. But here I'll be able to, right? We're going to spread out our action potentials here until they basically get down to about this same rate that we were initially at 0, right? So they spread out and they're sort of slowly getting longer. Maybe that's not the best lengthening of action potential. Maybe this one's a little bit further over, right? Maybe this one's more like this. But you get the idea that we're slowly lengthening our action potentials as we go down here. Whoops. Get rid of that one. Until we return to our base rate of firing, right? Okay. That's a tonic receptor. That's how that would behave. What about a phasic receptor, right? Well, a phasic receptor is not going to be doing anything during this period where there's no change at all. But here, where there's this rapid change, we're going to get a really rapid firing of this phasic receptor, right? There's lots of change. And really, I forgot to carry out my line here, right? Because, of course, this is, all these cells are operating at negative 70 millivolts. Here's our little hyperpolarization phase of all these action potentials, right? Okay. So, and our phasic receptor, of course, is doing the same thing. I'll just dot this line because it's also operating, in this case, at negative 70 millivolts, right? Okay, we have a really intense firing here. We're also going to have some firing start here, right? Whoa, wrong. There we go. Wrong color. We're also going to have some firing start here, but this firing is not going to be, they're not going to be as close to each other, right? They're not going to be nearly as close to each other because this rate of firing or rate of change of the stimulus is less than that rate of change of the stimulus that we had at the beginning. So yeah, they'll still be changing, or that rate is still changing. And so we're still firing action potentials, but not nearly as rapidly as we were here, right? Okay, so we get all the way down to this base rate again, or this base stimulus of 0, no stimulus, right? And then we get no firing of this phasic receptor any longer because the stimulus is no longer there, right? So this is the difference between these tonic, right here, and then these phasic receptors. Oh, that's what I know. These phasic receptors, right? This is really in terms of being always active, always firing, and tuned to the intensity of the stimulus versus, not firing until there's a change in the stimulus and the rate of action potential firing corresponding to the rate of change of that stimulus, right? You can see two different rates right here. Okay. Let's pop back over now to our Now you see that I've classified receptors by which type of stimulus excites them. Giving you some examples of each type. We've said this is and then I've also added whether their adaptation is rapid or not. So whether they're always on and don't adapt, or whether they're only on during changes and adapt quickly, whether they have a sensory structure or not, and then if we know anything about what type of neuron A or C or B carries this information. So nociceptors, painful stimuli. Again, nociceptors can respond. There are nociceptors that respond to all three types, and there are specific nociceptors that respond to only one or the other. These tend to have no sensory structure associated with them. So in other words, it's just a neuron with free dendrites, and the stimulus is acting on proteins in the plasma membrane of those dendrites, right? That's a free receptor. These are tonically active. We always need to know about this type of pain in general. We always want to know if pain, this is really thinking about acute pain. We always want to know about that type of pain. And so the neurons that are going to be carrying that information are typically, um, for terms of acute pain, they are large and myelinated, right? We know that these are the carrying information very quickly. In terms of chronic pain, right? They tend to be type C, unmyelinated and thin. We need to know about this pain, but it's not critical to our survival, right? And in fact, we edit a lot of this. If it's really chronic pain, we edit a lot of that out at the level of the thalamus. Chemoreceptors, they're responding to chemicals, even very small things like carbon dioxide and pH, which is really not a chemical at all. It's just basically hydrogen ions, right? Hydrogen ion concentration. These are also free. They tend to have no sensory structure associated with them. They're phasic, most of these. So they tend to, as long as it's not a painful chemical stimulus, these have rapid adaptation. Mechanoreceptors respond to some kind of physical pressure, deformation, tearing. We have a bunch of different types here that are interesting. We have a bunch in the skin, right, that tell us about what we're touching and what does it feel like. We have a bunch that are in like our blood vessels and in our heart that tell us about our blood pressure, how high or low is it, so that we can modify that. We have them in our lungs for the same, telling us about the pressure in our lungs. And of course, some of these receptors are actually visceral, right? Like we have here, these baroreceptors are visceral. And then we have proprioceptors these are composed primarily of free nerve endings. These are all the ones that we have in our joints and in our muscles. So we talked about muscle spindle um, receptors that are important for our reflexes, right? We also have those same types of receptors in our joints. So for as long as my arm is held out behind me like this, I will know about it because my proprioceptors are being pressured or stretched or compressed in the joint of my shoulder joint. Right? These are variable. Some are tonic, some are phasic receptors, but certainly the ones that are proprioreceptors, right, which are carrying information to our cerebellum very quickly. Remember, we talked about this in the brain lecture, right? Any that are carrying proprioceptive signals to the cerebellum, those are definitely going to be a type A, right? A very large diameter and myelinated, because that information is so important for us to move through the world and maintain balance as we do complex things like run and jump and ride a bike and all of that, right? And then we have thermoreceptors, right? Many more cold receptors than warm, about a 3.5 to 1 ratio of cold than warm, which just tells you that we've it's been important for us to feel um, various types of cold much more during our evolutionary history than it has been to feel various types of warmth. These are all free receptors, no sensory structure associated with them, and tend to be phasic, so they're fast adapting receptors. Now you can see the only one that really has a lot of um, that has a sensory structure, specialized sensory structure or cell associated with it, tend to be some of these mechanoreceptors, right? So we're going to look at those mechanoreceptors here a little more closely. These are tactile receptors. They tell us about position of the body, what we're feeling, what that feels like, how intense is that pressure. So for example, the fine touch receptors, these are called Meissner's corpuscles. They respond to pressure and vibrations of uh, like low frequency vibrations, like 50 up to 50 hertz. We have these, you can see here's stratified squamous epithelia and connective tissue underneath. They're right here at the apex, right, really close to the surface of the skin. These, so we'll see these in lab, and we saw them, I think, last week in lab. These Meissner's corpuscles. Um, these are phasic receptors. Um, let's do the other type in our skin here that are responsible for sensing deep pressure and vibrations up to about 250 megahertz or hertz. These are Pacinian corpuscles or lamellated corpuscles. They're actually much, much larger than these tactile corpuscles. You don't have a good feeling here, but look at this. This is magnified by 330 times the size of life. while this one is only magnified by 125 times the size of life, and it's already much bigger than this. So these are really big structures, but they're deep within the dermis. So they're far from the surface of the skin. They're very deep down into the dermis. So they're telling us about deep pressure, like you hear bass, right? That's really what's being stimulated is a lot of Meissner's corpuscles. Here we have some Ruthenian corpuscles. This is a free nerve ending. This is buried within the muscle spindles and also within joint capsules. So this is also sensing pressure, right? Pulls of collagen fibers. As collagen fibers get stretched, it changes the stimulation to these dendrites here. You can see it's got Schwann cells on here, right? You know this is important information. So these are large diameter axons and they're myelinated, and these are tonically active. We always need to know about this kind of information about where our body is and how flexed or contracted, flexed or extended, contracted or extended our muscles are, right? And then finally, here's some more that are associated with fine touch. There are some free nerve endings here. There are also some that have some specialized cells that respond to pressure attached to that sensory neuron. But these are more fine touch receptors responding to pressure. And these are tonically active, where these are physically active. So that's why, you can, if you do touch your fingers together, certain receptors are stimulated immediately and then stop sending information, like these Meissner's corpuscles, but others are stimulated and continue to send that information, like these free nerve endings here. So a variety of tactile corpuscles designed to do different things. And so whether they're tonically or phasically active really depends on how important it is that we get that information and have that information so that we can make decisions about what to do with it. And they do vary in terms of their distribution, very close to the skin, very deep in the skin. Here's more very close to the skin. And then those that are buried in muscle fibers or in joint capsules. Okay. All right. Thanks for watching this video. Please let me know if you have any questions about any of this.